All right, thanks everyone. My name's Tanya Applegate. I'm from the Surveillance and Evaluation Research Program here at the Kirby Institute, and I thank you all for coming here, joining us for those in the seminar room today at the Kirby and those joining us online. Before we begin, I'd very much like to acknowledge the traditional custodians of the unceded lands that we have the privilege of meeting on here today, and it's the traditional lands of the Bidjigal people of the Eora Nation. And I'd like to pay my respects to their elders past and present and welcome any Aboriginal and Torres Strait Islander people joining us here online in the room. So in the room and also online. And I'd also like to acknowledge the traditional custodians, all the amazing lands that you're joining us from today. So firstly, I just would like to premise this seminar with the fact that this is the final presentation for two PhD students who are in their final throes of their PhD. So Andrey Verich and Arthur Wong. And I will just briefly introduce each of our speakers, and they'll have about 20 minutes each. They've chosen different areas within their PhD, not necessarily the whole PhD, to present today to give you a flavour of the work that they're doing. And what I really like about the selection and the combination of presentations today is very much, I feel it's a great reflection of the sort of work that we do here at the Kirby, and in terms of the multidisciplinary approach, from the laboratory work all through to the public health and clinical impact work as well. So as I said, we'll have two presentations and each will have the questions at the end of each presentation. So for Andrey first and then for Arthur at the end. So please do feel free to put your questions in the Q&A chat. So firstly, I'd like to introduce Andrey Verich. So many of you will know Andrey. He's been at the Kirby Institute for quite a while, since he was a research assistant in the laboratory at the Viral Hepatitis and Clinical Research Program. He's currently doing his PhD in the Surveillance and Evaluation Research Program. But what's quite, I don't know if you knew this, Andrey, but he's doing an industry PhD, which is in line with the CSIRO in partnership with a private diagnostic company and also the University of New South Wales, so the Kirby Institute. And I think this is Andrey's the first one to take a foray into this sort of scholarship. There are other ones such as the International Industry PhD, and I think it provides this really nice connection between providing that opportunity to look at things from an industry perspective and to get experience working with industry and working with other external partners such as CSIRO. So Andrey, I think that's probably it for you. I'll hand over to you. Thank you.
Thank you, Tanya, for that introduction, and thank you for the acknowledgement. So, hello, everybody. As mentioned, I'm Andrey, and I want to talk to you about my industry PhD. And the snippet of work that I'll be talking about is how I've been looking at the data availability and performing machine learning for Neisseria gonorrhoea, whole genome sequencing, and resistance. So, as mentioned by Tanya, I don't think I have to go through this slide too much. Who am I? Well, I used to do a lot of microbiology-based work, diagnostic-based work, and now I'm doing this industry PhD, and the focus is on looking for diagnostic targets for Neisseria gonorrhoea resistance, rapid point of care tests. And I'm doing it through SERP, SpeedX, and CSIRO. So what exactly am I doing? So I'll be talking about the systematic review that has been done, looking for what data is available for Neisseria gonorrhoea whole genome sequence that is paired with minimally improved concentration, resistance information, and then use this large-scale data set to investigate for any information for resistance based on the whole genome sequence via machine learning. And then there's a third section where I'll be using an Australian data set that is highly detailed to investigate how patient information can influence the results of machine learning. However, for this presentation, I'll be focusing on the first two aims, which is the systematic review and the publicly available information machine learning analysis. Now, why am I doing this? Why the interest in Neisseria gonorrhoea? Well, because Neisseria gonorrhoea is a WHO party pathogen, it has exploding notification rates. That's the Australian ones. However, that mirrors most of the world, with the notification rates effectively more than doubling in the past 10 years. There are around 80 million annual cases worldwide. And the problem, of course, is that it has a high burden of disease. It can affect many different sites, primarily urogenital. There, it can cause inflammation, wreak havoc on fertility, and harm unborn children. So it's a big problem, and it's been set for elimination by the WHO by 2030. So let me talk about this systematic review, aiming to find what information is out there about this. So as part of the systematic review, I wanted to see how accessible whole genome sequencing information about Neisseria gonorrhea that's paired with resistance is, how often and what metadata about the sample, about the patient is present. And then I guess the question is, what and where is this data coming from? Because there's been a lot of discussion about if different anatomical sites might have influence on what kind of resistance profiles there could be for the Neisseria gonorrhea. And I wanted to see if this is something that can be investigated, what we have right now. This is because, as mentioned by the WHO for elimination, one of the other things they said is we need high quality and representative gonococcal AMR data, imperative to monitor AMR trends, etc. So I'm choosing to see this high quality representative data as data that's combining whole genome sequence information, resistance information, and then patient metadata. Because I wanted to see how this can influence my machine learning analysis, as mentioned before, and anybody else's. Because I'm choosing to perceive this as a potential self-fulfilling prophecy and Ouroboros of data will influence analyses, which can influence the policy, and the policy will influence what kind of data is being investigated. So I wanted to see at the baseline what kind of data is out there and how will this influence the rest. So the methodology of the actual systematic review. Here it is. What I chose to do was an individual patient data systematic review. In this case, what that means is I'm choosing to register single accession numbers of a whole genome sequence. That has to be present with a numerical resistance value that's laboratory derived. So at least one of these data points have to be present for a publication to be involved in the systematic review. And it has to be clinical isolates because I wanted to see where we're getting this data from in the world. I don't want to see a mutant from the laboratory, for example. This is the database that was searched over those years, ending in March 2024. Inclusion is at least one of those following antibiotics, had to have that numerical information. And I chose to investigate metadata at time of publication. Noting how we're nearing the 2030 elimination goal, I wanted to see how actively data is being released. So, however, when doing the analysis, I found that there was quite a lot of extra data that was available within the databases, Pathogen Watch, NCBI, PubMedLST. These are the ones that typically host whole genome sequence information. So I did a secondary supplementary analysis, we can call it, which chose to take any information from those databases at any time. However, the result you're seeing is what's available at time of publication, primarily, unless mentioned otherwise. So the important part, after going through the literature, after looking at all the duplicates and all the publications, we started around 1,600 abstracts, a lot of reading, down to around 62 studies that actually had at least one whole genome sequence with numerical resistance. Potentially, by taking the raw numbers from each publication, that's around up to 38,000 whole genome sequences that could be out there. The concern, 12,500 were accessible. This is a very large and immediate drop-off of what could be out there, with 66% of the data being lost between something that potentially is out there as useful information and then what is accessible for somebody to analyze, for example, myself with machine learning or phylogenetics, etc. Supplementary analysis was able to find around an extra 10% of the data, which means even after looking through the databases and what is available at publication, more than 50% of the data was missing, which to me is a bit of a concern. Then, now looking at the data, what is actually available at time of publication? The most common data was year sampled, which is promising to see. However, we're starting off with 66% of the data already missing, as mentioned before. So this is what's available in that other remaining percent. In that remaining percent, even in the most commonly sampled metadata, 30% of that was missing at time of publication. At least most of it being present is good to see. That it can be important for phylogenetics and tracking resistance as it develops. So that's good. Promising to see that most of the data is recent. So sampling is occurring right now, and that's being released active enough. However, there still appears to be a little time lag between samplings of the Neisseria gonorrhea and the reporting by publication, because noting that my cutoff was 2024, there was no data from 2023, 2024. Understandably so, there is a time delay between sampling and publication. However, small concerns with tracking resistance as it develops. Other point is there was a recent publication by Featherstone, I think, that was talking about how month-based information can be very important for improving phylogenetics. Only 30% of that data was present at time of publication, which could be concerning to make phylogenetics a bit better. The point is also with doing the supplementary data analysis, 10% of the year sampled was still missing. The concern here is because this data is meant to be a minimum requirement to input into those PubMLST and CBI and such databases. So even with a minimum requirement, 10% of this data is missing because somebody wrote in the word missing and that was accepted. So despite all the little attrition that's happening, even the most common data is missing 10% of the time, doing as much investigation as possible. Moving on from the dates, where is this data coming from? Because that will influence some of the results. Won't be surprised to see that most of this data is coming from high-income countries. These are the countries that have resources for the energy-intensive and staff-intensive processes, which is whole genome sequencing and MIC testing. Many of these countries also have surveillance networks in place already, so you won't be surprised to see that they're the ones releasing this information, with the United States, UK, and Austria being able to provide most of the information. I guess the concern here immediately is seeing where, knowing the burdens of disease of Neisseria gonorrhoea, with Southeast Asia, Africa having some more of the burden of disease, and then potential reservoirs of resistance, what we're not seeing is this information, the whole genome sequencing analysis. So a lot of this information can be missing. So there could be a lot of phylogenetics and a lot of commensal bacteria, sorry, not commensal, a lot of information about horizontal gene transfer that we could be missing if I am to do whole genome sequencing analysis. However, there appears to be an improvement with the WHO system called eGASP in place. Two of the publications were present in my analyses, which were about Cambodia and Vietnam. So it's promising to see that this system is in place and is beginning to alleviate this issue by presenting information from these countries. So this can improve potentially in the future. Now the resistance information. This is the weighted pooled global average resistance results, excluding case studies and reports, as they might bias the ratios of information present. And here, I guess the summary is, mouse is not seen. The summary is that for the not current last line antibiotics, the resistance is actually the average global. So that's promising to see. What is, I guess, also not surprising to see is that the current last line antibiotics have actually relatively high resistance rates with whole genome sequencing information. This is probably because of, again, with resources that are limited, what will people focus on? They'll focus on what's currently last line. Because there's a 5% alert value for being concerned about resistance, potentially ceasing treatment. And you can see cefixime, ceftriaxone, which are the current last line treatments. Cefixime is above 10%, ceftriaxone is close to 5% within this data set. However, we know that this is not the case. Ceftriaxone results, for example, are close to 1% or lower, actually, in global averages. So we can see that this type of information is oversampled when it comes to whole genome sequencing MIC. Now, to me, what was most, I guess, important, and one of my questions is understanding where this data is coming from in terms of population anatomical sites of sampling. Here is that data. Immediate concern, quite a lot of it is missing. You can see on the right-hand side, the collection site legend, and the bottom of the population category. Not recorded population, around 50% of this data is missing in the first place. Then, when looking at the collection site, this is the most common missing information. 75% of that information was missing, with most of it being NA. As mentioned before with the questions about where resistance could be coming from, our oropharyngeal samples potentially have high resistance for our last line treatments. This means that if I am to use this information for whole genome sequencing analysis, then it's going to be very limited. So it's concerning to see that as well. The other point about accessibility of this data, when having to process all this data, there was hundreds of different formats about how people would categorize urogenital, blood, etc. There's a lot of processing has to be done. So if anybody else needs to repeat this sort of analysis, it just complicates things more than necessary. As seen, when available, I guess unsurprisingly so, most of this information is from men, and urogenital and rectal oropharyngeal sites are undersampled. And this is probably from clinical attendants or people who have more symptoms coming from urogenital sites. So those are the ones that get sampled based on presentation to a clinic from men, primarily. Because knowing that there's a burden of disease that's probably equally shared 50% between men and women, it's concerning for me to see that around less than 5% of the data comes from women. We have some questions about if transcriptional factors increase different types of resistance profiles from cervix, etc. This means that this type of information cannot be investigated with these data sets effectively at the moment. That's concerning for me to see. So that's kind of a summary of the type of data that we're working with. from potentially 38,000 sequences, having less than half of it accessible, many of them missing metadata. And then when talking about that optimal high quality data, only 2.1% had information, for example, about sexual orientation, gender, anatomical site, etc. And I guess, is this concerning? Well, it depends on kind of analysis you want to do. I'll be talking about my machine learning analysis in a second. Much of that data can still be used, but I guess if you want to go into very highly detailed questions about what I've been mentioning, different sites, different populations, then it might mean that the results might be not conclusive. So I guess my question and statement is you got to mind the gaps, the geography, anatomy, population, because if you just take this whole genome sequencing information and just push it through a machine learning pipeline, you might be biased towards only urogenital samples, for example. It's concerning to see this type of data attrition as well, the huge drop-off that you saw before. And I guess it's not surprising that urogenital sites from men from high resource countries are the ones that are most commonly present. There's been some initiatives to fix this type of problem that I'm describing right now with the FAIR initiative, Findable, Accessible, Interpretable, and Reusable Data. So they've been providing guidelines and recommendations for databases and how to improve data accessibility and all those different aspects. And as always, very easy for me to request for people to share their data, but resources are limited with people, having to provide publications, do whole genome sequencing, and MIC testing, which are all very resource-intensive processes. But I guess just highlighting that, again, to reiterate, if any analysis comes from this, you got to make sure you mind those gaps because that can influence the results. Now, speaking of the analysis, taking that publicly accessible data, just ignore everything I said about the potential concerns of gaps and things. Let's talk about how I use that whole genome sequencing data resistance with a machine learning pipeline. So section 2 of this presentation is about the whole genome sequencing analysis pipeline for machine learning that I've been developing with the help of a lot of tools made by CSIRO, the partners of the industry PhD, because the goals are very simple. Develop a pipeline and utilize it to look for gene variants that could be of interest for resistance, gene panels, etc., for surveillance and diagnostics. Now, why? Why does this need to be done? CSIRO gonorrhea has a very highly variable resistance. We're seeing there's the most common different antibiotics, the most recent one, sorry, targeting different components of CSIRO gonorrhea. I'll be talking about a bit more CSIRO here. And the concern is CSIRO gonorrhea is very good at getting that resistance. Many different antibiotics have been tried. Many different antibiotics have been abandoned because it's been able to get resistance from other Neisseras that are commensal in the human body and or just develop it through plasmids and such. There's a lot of different complications with the whole genome sequence information. This is why I think something like the machine learning approach can be very important because it's handled to design a lot of different information. And this is a lot of different potential information. The whole genome sequence of CSIRO gonorrhea is around 2.2 million base pairs. That's 2.2 million data points that can all influence the outcome. And machine learning is something that can investigate that. So the actual methods. Taking the previous information that I was talking about before, minimum inhibitor concentration and whole genome sequence, that was prepared for this pipeline. The resistance data for this pipeline had to be split into resistant or susceptible. Data balance is important for machine learning, which I guess is a highlight for the previous data that I was talking about before. And I chose to use the UCAS breakpoints, combining susceptible and intermediate into one category and resistant into its own. And the whole genome sequencing information, that was processed by taking a whole genome sequence of the CSIRO gonorrhea and aligning it to a reference. The reference that I chose to use is the following, the WHO F chromosome. That is the largest reference genome for CSIRO gonorrhea, whole genome sequence. It is also susceptible, so as variants, it will be interesting to see because they're most likely to be contributing to resistance. I also chose to input some plasmids. These plasmids are some of the ones associated with resistance right now. And there's the cryptic plasmid, nobody knows what it does. So I'll just say I just chose to put that in just to see what happens. Simple as that. Now the actual machine learning pipeline. Here I'm using the program called VariantSpark. That's been developed with the CSIRO. This does analysis to determine if something's contributing to susceptibility or resistance based on the variant call format and the input of the resistance profile. How does it work? It uses a random forest algorithm, which effectively is like a team of experts that is single point there. They get presented a random selection of variants, knowing which variants are present from which samples, then they will determine which samples are the most important for contributing to resistance. This team of experts gets duplicated hundreds of times, gets presented many different random data sets, and then the average of their most important selection is taken to contribute and determine if this is most commonly picked, it's probably the most important for contributing to resistance. All these parameters are highly adjustable. I will not be going into the details about all the depth of trees, the amount of decisions they have to make, how much information, different types of data, variants are presented at each node. And Variant Spark is designed to be, as I'll call it, a self-validating machine learning process. Effectively, what that means is, as mentioned before, any information not randomly presented for that tree or team, whatever you want to call it, is then fed back to that node. And based on the information assembled through the consensus, will they correctly determine if that sample with those variants will have resistance or not? So effectively, it takes the information was not presented and knowing that the result is susceptible, will it correctly predict that. The aim of doing all of that, going for all this machine learning business, is to avoid what you're seeing here. Because this is the, we can say, raw results of the analysis. What you're seeing on the left, y-axis is the importance value, effectively. X-axis, bottom, is the whole genome sequence length of Neisseria gonorrhoea. Those plasmids are present on the right with the different colors. And each one of these dots is a single variant along the whole genome sequence with associated importance. That is, for example, present there. This is logistic regression. This is an older method of machine learning, and this is what I want to avoid, because this is how it now looks like if I'm using the more developed pipeline using Variant Spark. Those are meant to be the same data, so that is the same data, but these are the different analyses methods, and you can immediately see how improved the signal-to-noise ratio is. So when looking for diagnostic and/or resistance targets, you can immediately see that JARA is highlighted as a very important variant, which is promising to see, as that JARA variant is the one that's right now used for some diagnostic tests. So it's kind of validating the importance of that result. What I don't have time to is go into every single one of these data points because there's a lot of interesting things. What I can comment on is quite consistently housekeeping gene, tRNA synthesis genes have come up as important results for these analyses. This is promising to see as the genes that I'm highlighting there, which ILS-TGT MyAb, they've been found to contribute to superfloxacin resistance as well. And for example, E. coli, and this is the first time they're being potentially implicated for Neisseria gonorrhoea as well. Although, as you can see, not as important as JARA. Now, the pipeline, there's multiple different steps. Knowing how resistance can come about from multiple aspects, it doesn't have to be just a single gene. It really actually is a single gene like JARA over there. But I wanted to use this pipeline to see what other interactions from variants could be present to contribute to resistance. So another program was used called BitEpi. That was also made with CSIRO, and that BitEpi program effectively looks at if samples of variants are present together at the same time within the data set from the random forest algorithm. So it takes the important results from the random forest before, the Variant Spark, and assesses if they're commonly present together. And effectively looks for if they're present together in up to groups of 4 in this case. What was most promising to see, again, was seeing how JARA S91F, the known resistance target, came up as a single important variant for resistance. However, other important variants were highlighted as well with that constellation you're seeing there. The gray is a single variant, the equivalent of a single dot from the Variant Spark results. And the greens you're seeing there are 4 variants interacting, potentially contributing to resistance outcome, a phylogenetic outcome. And what this tells me is that now, if you want to take this result, validate it within the lab, etc., what you want to do is combine those mutants together at the same time. Because, for example, if you wanted to look at them one at a time, you might miss out on potential information and the importance of those variants together. Now, just in a moment, other resistance results. Very promising to see how known variants came up as well with efflux pumps. Don't have time to go into details. This is for azithromycin. This is for tetracycline. And I guess it was promising to see how the plasmid, despite a very small genetic structure, I think around less than 50,000, was able to come up with a lot of important results. So it's nice to see how Variant Spark was able to operate with this, because there was an initial concern if it will be able to operate with such small data sets. And it also came up with RSPJ, which is a known resistance target for tetracycline from the chromosome. So it's nice to see that as well. I keep seeing the tRNA synthesis genes, but we will not get into that at the moment. So conclusions. That's a quick run through of talking about the superfluoxetine results primarily, talking about the machine learning pipeline. Now, a lot of things can influence these results. As mentioned, reference selection, it's a variant call file. So if a gene is not present in the reference, it will not be present within the variant. So it's very important about what gets chosen for that. Machine learning parameters, breakpoint system for calling susceptibility or resistance. Also, other point, there's been multiple other steps I didn't have time to mention here. For example, there's a step that was also designed, RF local FDR by CSIRO, that takes the information from the random forest. Technically, it does not have p-values, but that's able to assign p-values of importance, and that gets fed into BitEpi. Principal component analysis for machine learning, protein simulations. I used to do a lot of protein work with my Babs, so I wanted to use machine learning, and that is a visualization of the mutant of JARAS91F, but I'm not going to go into those details at the moment. And as mentioned before, right now there's development about using Australian data sets which have highly detailed information, because the problem before was highlighted how often it is missing, to see if these patient factors will influence some of these results, being fed in as additional parameters into this machine learning model pipeline. And of course, it's nice to talk about all of this dry lab work, we'll say, but as somebody who used to do a lot of lab work, ultimately all of this does require lab-based validation. But I guess the point of all this analysis is to see how much computer work can be done before being required to validate it in the lab. Because, for example, you saw the logistic regression result. If you took those raw results, you'll never have enough time to go through hundreds of different mutation studies. Finally, last bonus point, machine learning. I had to learn it for this PhD, but during the process, I found that anybody can do it now. There's a program called Orange, which is a visual user interface. So if anybody's interested in trying machine learning, you can literally drag and drop things like there and use your data set, and it will give you many different models. There are some basic limitations, but really, to be honest, it's a good way to get into it if you're interested. I'd like to thank everybody who's contributed to this, my supervisor, Tanya, and everybody else, co-authors, and the AMR Hub. Thank you.
Thank you, Andrey. It blows my mind that Andrey taught himself all of that off the bat from the beginning, coming from the laboratory and then working on his experience on sequencing, then applying it to another completely different field in machine learning. So are there any questions from the audience here? Look online. Surely, Brendan, you've got one. No? Yes. Curtis.
Yeah, a great presentation. I was curious on the point about all the missing data at the start and then kind of thinking about the machine learning model in the second half. Is there maybe like one or two really specific data points that you wish you had that you want future data sets to include to update your analysis? Is there any like one kind of, that's it, that's the thing, if I had, this thing could be way more accurate?
I guess for whole genome sequencing, the data is always there, but the anatomical site, I think, is very important because of the current debate, oropharyngeal, anorectal, do they have different resistance profiles? Will they have different outcomes? Knowing if that's present from what site, then that can be also input as a variable, because you can put any binary variables, whatever, preferably binary variables, into the machine learning model. It'll tell you it's not that variant that's important, is the fact that if this is an anorectal sample that it has resistance, like it can do that as well. So that can be important just to see, is there a difference? Because right now it's still a hotly debated topic with different papers saying oropharyngeal resistance more, others say no. So the more data, the better to determine that.
Any other questions? There's just one other comment. It's not a question. It's so you're probably off the hook. It's just a reflection on the EGASP that you mentioned as well, which is enhanced gonococcal surveillance program. And they are expanding over time. They've been expanding the number of countries to support what you acknowledged, which is the fact that high income countries may have the resources to do the sequencing and all the culture work, which is the minimum inhibitory concentration that Andrey referred to. But the lower income countries don't, and that's where a lot of the burden is. So EGASP has over time been bringing countries on board with being able to do this work. And it's obviously very expansive, very time consuming, and to make sure that the quality is high with support of our colleagues such as Monica Lara over the road at the WHO Collaborating Centre to support countries develop this not real time, as you've noticed, but the high quality data. One thing to acknowledge is that that was all at risk with the changes to WHO funding and the cuts. As you know, they've completely had to restructure and they've lost a significant proportion. Thankfully, somehow they managed to squirrel away the funding. I say squirrel away, I say that, and that's not, they protect the funding. So that work will continue. So hopefully you'll see more and more data coming through from the countries that bear the greatest burden as well.
Yes.
Are there any other questions? If not, I'd like to thank Andrey and so I'll move on, and I should have done this at the beginning, sorry, Arthur, but I'll do it now to introduce Arthur Wong, who many of you may have met. Arthur, who was introduced to the Kirby family probably 14 years ago in doing his honours and as part of his medical training, and then went overseas to continue his specialisation, returning to Australia now, being a sexual health physician in STI and HIV at Sydney Sexual Health Centre. He's also head of what is called Teaching and Evidence at the South Eastern Sydney Local Health District, and that's another name for head of research. So we're really grateful for being able to continue to expand that collaboration with regards to the clinical questions and the clinical applications of these diagnostic cohorts. And also just like to, when Arthur came back into our world and said that he was keen to explore doing a PhD here at the Kirby. And I said, first thing I said, you need to meet Nick. And those two bonded, as you imagine. We're very grateful to have had that opportunity to have worked with Nick as part of this project. So I'd like to welcome Arthur. Thank you.
Hi, everyone. Thank you. So yeah, I'm Arthur. I'm one of the PhD students at SIRP, and I also work as a doctor in Sydney Sexual Health. And my talk is titled, Are we overprescribing antibiotics for chlamydia and gonorrhea in men who have sex with men? I would also like to acknowledge the traditional owners of the land on which we're meeting today and pay my respects to elders past, present. Just upfront acknowledgement. So thank you to my amazing supervisors here at the Kirby who have really shaped my PhD journey. I also received a scholarship from the AMR Hub, and a lot of this work that I'm about to present is generated in collaboration with co-authors at the Hub as well. So just briefly about background about chlamydia gonorrhea in New South Wales. So unsurprising to all of us here, we're still experiencing quite high rates of notification in the state. In particular, in gonorrhea, the rates are still rising quite rapidly. And we have quite a high male to female preponderance for gonorrhea in the state, mostly because the prevalence is much higher among MSM. So unsurprisingly, MSM remains a key priority population in the state's STI strategy. And where I work, a publicly funded sexual health clinic is a key setting as a place that we respond to this epidemic. Just a really quick overview, again, probably not new information to many in the audience, but in general terms, there are two main ways that we test for chlamydia gonorrhea in people, and we give antibiotics. So for example, sometimes people come to clinic and they have symptoms that may be suggestive of chlamydia gonorrhea. We see them, we might do some tests like microscopy, and we make an educated guess about which infection they may or may not have. And with that information in mind, we give them what we call presumptive treatment. So we give them antibiotics to cover what we think is present. But at the same time, we also send some confirmatory tests in the form of these highly sensitive nucleic acid tests, just to see what pathogens are in fact present. Alternatively, we also ask certain at-risk populations to come back frequently for asymptomatic screening. So these are people who have no symptoms, but they come back every 3 months. For example, MSM on PrEP are asked to come back every 3 months, and we do a screening test in all 3 of the relevant anatomical sites, so throat, rectum, and urine, looking for chlamydia gonorrhea, again, with these really sensitive nucleic acid tests as well. And if they test positive, obviously we let them know and they come back for treatment. This paradigm is obviously a really oversimplification because I haven't included scenarios, for example, if they received a point of care test, for example. So how would these pathways potentially lead to unnecessary treatment? So for example, in people with symptoms, we give them presumptive treatment and we do tests. Often after the fact, we find out that actually, in fact, they've tested negative for one or more of the pathogens that we've covered them for. So you could argue that we've given them a necessary treatment. On the other hand, in people that are coming back for screening, even though we might find some of these pathogens on these highly sensitive nucleic acid tests, we know that at least a small proportion of them are in fact non-viable bugs. So we find them, we find the DNA, but actually they might be just a bit of transiently deposited remnant nucleic acid from recent sex, or they're bugs that have been spontaneously cleared. But in the current paradigm, they will still get treatment, which is arguably unnecessary. So why is it a problem to be giving people unnecessary antibiotics? So as Andrey was mentioning, both in the WHO academic literature and in the lay literature, resistant gonorrhea is becoming a huge concern. And there's more and more evidence that if you look at populations that undergo frequent asymptomatic screening for chlamydia gonorrhea, for example, in cohorts of patients who are undergoing PrEP follow-up, you do find that the more you screen, the more likely you are to find the emergence of resistant gonorrhea isolates. But for the sake of balance, it's also really important to mention that even in settings where there is no intensive longitudinal STI screening, you will also find emergence of resistant gonorrhea. So although over-screening and therefore over-treatment may be part of the reason, it's certainly not the whole picture. But with that in mind, this particular issue has taken an urgent tone in New South Wales, at least this year, because although fortunately we haven't had many very resistant gonorrhea isolates historically, this year for the first time we've had a few cases of both locally acquired and imported cases of extensively drug-resistant gonorrhea isolates. So with that in mind, in my candidature, we asked 3 research questions. The first one is we wanted to know what the quantities and the indications of the antibiotics that we're giving in Australian publicly funded sexual health clinics are. We wanted to try to estimate what proportions of presumptive treatment for symptomatic MSM is potentially unnecessary. And we wanted to find out what proportion of screening-related treatment in asymptomatic MSM is potentially unnecessary. And I'll take you through each one of those one by one. So for the first question, we looked at the amazing Access database. For those of you who may not know, Access is a very large collaborative project where both public and private sexual health clinics feed in data anonymously about testing and treatment and kind of surveillance information about STIs and HIV. And for this particular project, we went on the basis that we knew that widespread PrEP implementation and therefore the associated recommendations for longitudinal intensive chlamydia gonorrhea screening really came about kind of between 2016 to 2019, where PrEP became subsidized. So then we went back and had a look at all the electronic medical records during this period, right up to 2023, from all the publicly funded sexual health clinics among MSM who went to the clinics for screening. So these are people who turn up at least once a year for some asymptomatic screening visits. And in a cohort group level, we stratified all their testing and treatment indication to either screening related or non-screening related. And ceftriaxone was the antibiotic that we chose to focus particular attention on because of the fact that, as Andrey mentioned, is a last line antibiotic for gonorrhea treatment. But on an individual level, we also constructed a multivariable model to look at the relationship between how often someone screens and how many doses of ceftriaxone they're likely to receive in a calendar year. And for simplicity, I will truncate all the non-screening related testing and treatment into just one category for this presentation. So this is the first of the findings. So if you look on the left-hand side of the panel, from the beginning of the study period to the end of the study period, the kind of per person year prescription rate of ceftriaxone has almost doubled. And what's particularly interesting is that if you look at the indication for which the ceftriaxone was prescribed, there has been a bit of a reversal. So although at the beginning, a lot more non-screening related ceftriaxone was being given, by the end of the study period, actually, it was the screening related ceftriaxone that was a main indication. But it's also important to notice that throughout this period, the volume or the per person prescription rate of non-screening ceftriaxone has remained quite high as well. So the total volume is increasing. So in this first bit, we found out that, we're giving a lot more ceftriaxone in MSM who are undergoing gonorrhea screening, and then the leading indication for prescription has also changed. On an individual level in the multivariate model, we found that even adjusting for covariates that predicts kind of the risk for acquiring and being diagnosed with gonorrhea, there remains a dose-dependent relationship between the number of screening tests someone undergoes and the number of doses of ceftriaxone that they receive. So the next part of the next question we wanted to find out was about presumptive treatment in symptomatic people. And to answer this question, we conducted a retrospective analysis looking at one particular clinic, in the clinic that I work, and in this analysis that's been published last year. So we looked over a 2-year period at a single center, and we looked at all visits involving symptomatic MSM who came to us for presumptive treatment. So these are people who got treatment at a time when their STI results were not known. They had to have undergone testing on the same day as treatment, and also they didn't receive any antibiotics in the preceding month. So these are people that we definitely were only treating presumptively. So looking at these people, so at the end, we found around 1,000 visits, and looking at these people who got treatment, we found that actually around 40% of them ended up not testing positive for either chlamydia or gonorrhea across all 3 of their anatomical sites. Again, we pay particular focus to ceftriaxone because of its significance, and we found that among almost 600 doses of ceftriaxone we've given to these patients, around a third of them were given to people that were subsequently found to be gonorrhea negative. And if you stratify the cohort into the kind of symptoms they presented to us for, actually, if you look at people who came to us with urethral symptoms, it's quite unlikely that they got unnecessary ceftriaxone. But if you look at the other symptoms, particularly for anorectal symptoms, actually more than half of them got unnecessary ceftriaxone. Next, we constructed a multivariate model to look at what factors are associated with getting unnecessary ceftriaxone. And unsurprisingly, again, it's those particular symptoms that were associated with getting unnecessary ceftriaxone, but also other patient characteristics that likely increases the pre-test probability or clinician suspicion that someone has gonorrhea. So if someone told us that they had gonorrhea recently or they were in contact with gonorrhea. This is after we have adjusted for other factors that may be associated with high risk of STI, such as partner numbers, condom use, and PrEP use. The other part of this paper was that we wanted to see, because currently in the current clinic models, we already had some rudimentary forms of point of care testing. For example, if someone has discharge or secretions, we could look under the microscope for gonorrhea, and people who presenting with anorectal symptoms, we can perform a procedure we call anoscopy, where we have a look inside the anorectum just to find any evidence of gonorrhea. So we want to see if patients have undergone these procedures, are we more likely to prescribe antibiotics more precisely? And reassuringly, in people with urethral symptoms, indeed, if they had undergone microscopy, they were much less likely to have received a necessary ceftriaxone, which is good, so that works. But disappointingly, in the other symptoms, even if a patient had undergone microscopy or anoscopy, they were not any less likely to receive a necessary ceftriaxone. So for this question, we found that a lot of ceftriaxone we're giving for gonorrhea coverage in symptomatic MSM is unnecessary. And therefore, we really need better tools for us to have more prescribed and more precise antibiotic prescribing and improved stewardship of antimicrobials. And often in this kind of discussion, molecular point of care testing is often raised as a solution. And indeed, there has been some compelling data out of Melbourne that shows that in a clinic setting, if you have molecular point of care testing available, you will reduce the amount of presumptive unnecessary treatment and overtreatment. However, for this intervention to be scalable and more widely adopted, we really need faster turnaround time. So ideally under 20 minutes of these assays, and for it to be affordable so that more clinics can afford to implement them. Okay, for the last bit of my talk, I'm going to touch on asymptomatic screening. Again, probably known to many of you in the room, in recent years, there has been a pretty intense debate about the benefits and risk of frequent asymptomatic screening of chlamydia gonorrhea in cisgendered MSM. And it's quite a complex debate. So for this talk, I'll really hone in on a particular strand of the argument, which is the idea that some of these pathogens that we're detecting on screening with nucleic acid testing are in fact not replicating and not viable, and therefore they actually pose no risk of transmission or health consequences. And therefore, if we treat all NAT-positive chlamydia gonorrhea, some of it will be unnecessary. So we actually started with this question with chlamydia as the pathogen of choice. The reason for this is because chlamydia actually already had quite a few research use only assays in existence that could tell us if it's viable or not. So for example, you can try to culture chlamydia. So if chlamydia is grown in the lab, you can replicate it, then it's probably viable, although the assay itself is quite insensitive but highly specific. Alternatively, you can try to stain the only infectious and replicating part of the chlamydia life cycle. So if it's positive, you can infer that it's viable. You can also look at transcribed messenger RNA. You can also look at membrane permeability of chlamydia. So if something has an intact membrane, you could infer that it's a viable chlamydia or vice versa. And lastly, you can use kind of concurrent real-time PCR test to look at the ratio between expressed RNA and genomic DNA of chlamydia to see what ratio is more suggestive of viability. And because we already had all these tests in existence in the literature, we performed and published a systematic review last year where we looked at any existing studies essentially that measured chlamydia viability. So in the end, we found 16 studies that were eligible for analysis, and we stratified the proportion of non-viable chlamydia found in these tests, and these papers, according to anatomical sites. And although this is a talk that's predominantly focused on cisgendered men, what we found was quite interesting in that if you looked at urogenital chlamydia that are found on nucleic acid testing, actually only very small numbers of them were non-viable. However, if you start looking at extragenital sites like the rectum or the throat, that's where you find higher and higher proportions of non-viable chlamydia that is being picked up on nucleic acid testing. And it is precisely these extragenital sites that we are undertaking frequent asymptomatic screening in MSM at the moment. So we found that, at least in the existing literature, there is evidence to support that a significant proportion of extragenital chlamydia that is currently being detected by NAT is actually non-viable. Next, we moved on and we wanted to find out, answer a similar question for gonorrhea. The problem we encountered here is that unlike chlamydia, there aren't a lot of existing viability tests for gonorrhea besides culture. So culture in the conventional sense is quite an established test for gonorrhea, but just like culturing chlamydia, it can be lacking in sensitivity, especially if you haven't collected or transported or incubated the gonorrhea in the appropriate method. So you could have false negative viability tests if you use culture alone. So there really need to be a new, more reliable, ideally molecular-based test for gonorrhea viability that can tell us in a reasonable time frame whether NAT-detected gonorrhea is viable or not. And we're very fortunate that our industry partner SpeedX had actually developed a novel assay they call Insignia, which is essentially using two concurrent real-time quantitative PCR reactions to measure both total nucleic acid and expressed RNA from gonorrhea. And using the difference in signal of the two PTR reactions, they could calculate what they call the viability index. So if the viability index is above a threshold, then it's determined that the gonorrhea infection is viable and vice versa. So in collaboration with SpeedX, we conducted a pilot proof of concept study in the clinic, trying to see whether we can use this assay to determine viability of gonorrhea. So in this study, we pragmatically recruited cisgender men with both suspected and confirmed gonorrhea infections in the urethra or the rectum. During the study visits, we collected 3 samples from these participants. The first is that we collected a standard commercial NAT test for gonorrhea in the form of the COBAS. And then we collected 2 additional samples, the first being a gonorrhea culture test that we transported in an enhanced manner to increase the yield of the culture as much as possible to use it as a comparator for viability. So we transported these gonorrhea cultures in special medium and in CO2 enriched environment. Lastly, we also collected a research swab that we sent to SpeedX, which they used this research assay insignia on, and also did a second confirmatory nucleic acid test to ensure that the gonorrhea is still present and detectable by a second assay. And because this is a pilot study, all participants received antibiotics if they were found to have gonorrhea with the conventional assay. And I'm going to focus particularly on the result of rectal gonorrhea because it's more interesting. So in the end, we recruited around 33 people that had rectal gonorrhea infections that were found on the COBAS assay, which is the standard of care. Among these, 29 of them had rectal infections that were confirmed by a second commercial PCR platform, the Resistant Plus GC, and which means that these people fulfilled the strict laboratory definition of extragenital gonorrhea infection according to the Australian lab definition. Next, we applied this research assay insignia, which was able to detect 27 out of the 29 confirmed gonorrhea rectal infections. Among these, 25 of them were actually determined to be viable, and the vast majority of them were positive on culture. One sample was found to be non-viable, and that was culture negative, and one sample had a viability index that was on the cutoff threshold value, so that was indeterminate, and that was also negative on culture. And at this point, it's really important to mention that we actually ended up recruiting quite a large proportion of asymptomatic people in terms of rectal infections. And the median number of days between their study visit and the last reported sex was around 2 weeks. So what did we find in this final bit of the talk? We found that in this cohort of patients that are predominantly asymptomatic and had rectal gonorrhea, that around 1 in 27 of them had detectable but non-viable gonorrhea, and 1 in 27 had borderline viability, and both of these people had negative culture. And although obviously this is a pilot study and probably too early to tell, there's a possibility that they were treated unnecessarily if they had actually non-viable organisms. And we have planned further studies where we will look more into these people with kind of low viability threshold using additional assays to tie break if there's a discordance between culture and viability tests. So I'm just going to wrap up. So overall, we found that we're giving a lot more antibiotics to MSM since we've implemented more intensive longitudinal screening recommendations for gonorrhea. But also the rate of non-screening antibiotics has remained quite high. Among symptomatic MSM, giving them presumptive treatment has led to quite a significant amount of unnecessary antibiotics, and it's more than half if you look particularly at ceftriaxone for gonorrhea coverage in rectal symptoms. What's the solution here? It's still up for debate. Some proponents are proposing that we should implement molecular point of care testing more widely in more clinics. Others would argue that we should reorientate our services so that patients with mild symptoms should get tests first and only treated if they test positive on that. Among asymptomatic people, there's a lot of evidence that screening is leading to giving unnecessary antibiotics to people with non-viable infections. And this seems to be a particular problem in extragenital site, like the rectum, where up to a third of chlamydia we think are non-viable. Again, what's the solution here? Is it to stop screening altogether, like some jurisdictions have implemented? For example, in Belgium, in the latest PrEP guidelines, they have actually stopped recommending frequent chlamydia gonorrhea screening among people who use PrEP. Alternatively, is a solution to give more technology to use reflex viability testing, implementing in the screening program to increase prescribing precision? The jury is still out, and a lot more evidence is needed to settle this debate. So for that, I would like to conclude and just thank again to all my collaborators who have helped me with these projects and just share this beautiful photo of where I work at Sydney Sexual Health. Thank you.
Thank you so much, Arthur. Incredible foray into some of the work that you've been completing as part of your PhD. Are there questions from the audience? Chan.
Hi, great talk, Arthur. I'm quite interested in the non-antibiotic treatment options, and like where is that at in terms of clinical approval or research?
Yeah, it's a good question. So I mean, in a sense, currently we already exercise a level of judgment. Like if a patient comes to us and say, I have mild symptoms, I'm really worried about taking unnecessary antibiotics, because, in the lay public press, there's also a lot of concerns about that. We often have these discussions and we explain the pros and cons of getting everything now versus testing coming back. And obviously it depends on the person's circumstances. So in terms of like, what's an approved clinical approach, in a way, we already do that. But we just have to negotiate on a case-by-case basis with patients. I suppose what some people are proposing is, rather than the dial leading heavily towards giving people presumptive treatment unless they opt out, it might be the opposite, where we say, if you have mild to moderate symptoms, we usually test first. Unless you're really concerned or you're not able to come back, then we can still offer you presumptive treatment. So it kind of, I guess, in that part of the proposal, it's wording the guidelines slightly differently in such a way that we give clinician a bit more autonomy in terms of deciding, not necessarily having to offer presumptive treatment to absolutely everyone. Because at the moment, at least in Australia, that's how the guidelines word it. Like it reads as if no matter how mild the symptoms are, if you're worried that they have rectal infection, you should give them everything, even if the patient's happy to come back later.
Perhaps I've got a follow-up question, but it's a bit more around the acceptability from a patient's perspective, some insights from our local, from a local Australia perspective. So you've mentioned that in the lay public news that people are aware of overprescribing and are more cautious now about using too many antibiotics. What about where do people sit with the treatment of asymptomatic here in Australia? And you mentioned that Belgium already was it Belgium that's has moved away from treating asymptomatic. Do you think we're going to head that way? I said there's more information needed, but which way do you think we're leaning at the moment? And even from a patient's perspective, would that be an easy sell, for example, if we did go down that pathway?
Yeah, great question. So I think it's a new frontier that sexual health medicine is going to, in terms of not testing so much anymore, because I think we've gotten really comfortable with testing a lot. There's been some really good kind of social sciences data out of Victoria, more from the clinician perspective about clinicians will be very uncomfortable not treating a patient if someone's test result is positive. So I guess the take-home message from that is, no matter what policy pathway we decide to go down on, it will likely be in the form of not testing, not treating, because I think all of us would be quite anxious not treating if it's positive. So, for example, in the European jurisdiction that have stopped screening, the public messaging has been that things like chlamydia, gonorrhea are natural and transient bugs in some people who have sex. If you have no symptoms, because they self-resolve, and if you have no symptoms, they don't cause any harm, then we just don't test for them. But if you have symptoms, then come back and we will test and we will treat you, and the treatment will help you feel better. So that's kind of at least in the Belgian experience, that's they've distilled the quite complex scientific messages into a few public-facing messages that they've used to kind of, I guess, shift people's thinking about the role of frequent asymptomatic chlamydia screening in this example.
I think then, do you think the concepts around viability testing and the messaging around there and the uptake utility of that might be also a simple message or a complicated message? So as you've mentioned just then, from my understanding, is that you're stepping away from testing, because if you test, then you want to do something about it if you get a result. Viability testing, I imagine, will give an even more sort of complex result to manage. How do you think viability testing is going to sit into the future? Do you think it's something that would get taken up? How do you think it would get used in a clinic?
Interesting question. So yeah, I think that actually is still a bit of work to do about the natural history of chlamydia, gonorrhea infection. I think to allay anxiety from clinicians about under treatment, I think we'll probably have to produce a lot more evidence about the accuracy of viability testing before we can more widely implement it. Yeah.
Okay. There are no questions online. Are there any more in the online? Please let us know. No, too slow. Virginia, antibiotics. Not your question.
No, that was a great presentation. Sorry, I missed some of it, but I had two questions. One was about a vaccine. This is a new area for me, so I'm just wondering what the latest is in that area. The second thing is that we we've done a number of studies looking at antibody use in the region, Asia Pacific, in across a range of areas, TB being a particular focus. And a lot of it is about patient demand. It's not only, but in these settings it is. And I just wondered about the patients that are receiving antibiotics. Are you able to, do you have information about their follow-up? Because one of the challenges for us has been that people may not need it, but they receive it, and sometimes they feel better and they go off and, well, was it needed? Probably not, but it hasn't actually necessarily done any harm, and people in their minds feel it's worked for them. So I just wondered what follow-up of patients that did receive an antibiotic, what's sort of their outcomes?
Great question. So I think the vaccine is a very complicated one, and I think this is an area where our understanding about natural history of these infections need to catch up, because there's been I guess a bit of a precedence where if we use really highly sensitive nucleic acid testing only as an endpoint for the efficacy of new interventions, there's a risk of underestimating the efficacy, because it may be that vaccine protects you from viable infections, but you may, if you do reproduce these vaccine studies, you might find a lot of people have non-viable but positive NAT test, and so in a per protocol manner, you might be underestimating the efficacy. So that's one proposal, is like, even if we don't widely implement viability testing in routine clinical practice, can we use it in things like therapeutic trials or vaccine trials? I don't have a good answer, but I think it's really important that we don't miss if vaccines protecting against viability of infection versus NAT positivity. In terms of follow-up, it's interesting because historically, although we've moved away from it because of resource constraint reasons, historically, the best form of follow-up we've done with STI testing is test of cure, which we don't do routinely anymore, except outside of a study setting. But when we read about like why do we recommend ceftriaxone as treatment, is because in the studies we've done test of cure, and 97% of people are NAT negative. But actually, we very infrequently ask patients who may not have positive results whether they're feeling better even though they got antibiotics. So it's a really interesting question, but I don't have a good answer for that. Yeah.
Thank you.
Thank you.
Okay, thank you. We're over time, but I'd like to thank Andrey and Arthur once more for sharing their PhD work with us. Thank you very much.